everybody. So it's my great honor to introduce the winner of the 2024 Lifetime Achievement Award. And this is awarded jointly by the Society for Developmental Biology and our official journal, Developmental Biology, in recognition of our long partnership in dissemination and promotion of the field. Now, this is an award that goes to a senior developmental biologist in recognition of their outstanding and sustained contributions in the field, exceptional mentoring, and service to the scientific community. And I'm absolutely delighted to introduce this year's winner, Dr. Deborah Andrew from Johns Hopkins University School of Medicine, because she perfectly embodies these characteristics. Debbie Andrews had a truly inspiring career as a developmental geneticist, and I'd like to share a little bit of her personal history to start with. So she's risen to be the Bayard Halsted Professor in Cell Biology at Hopkins, having started out as a first-generation student, moreover one who lost her father when she was just 4 years old, and then when only 23, lost her mother to breast cancer. Now this left her as the primary caregiver for two younger half-sisters, who remarkably she took with her to graduate school, raising them while working towards her degrees. She began her undergraduate studies at the University of Florida in limnology, but took a fly genetics course with David Kuhn that led her to pivot to the study of fly developmental genetics, pursuing first a master's in the Kuhn lab, and then continuing on to PhD work with Bruce Baker at UC San Diego. It was as a postdoc with Matthew Scott, first at Colorado and then Stanford, that she discovered a role for the Hox gene, sex chromosomes reduced, in salivary gland development, a finding which helped set the trajectory of her future career. Debbie joined the faculty at Johns Hopkins in 1993 and has dedicated her independent career to studying the developmental genetics of organogenesis in Drosophila. And these beautiful images I stole from her website. I'm sure we're going to see many more in her talk. Her nominators shared that, quote, her studies on organogenesis have defined the paradigm for how cells coordinate their activities to form functional tissues and organs. Her beautiful papers explain how cell fate is specified in the early embryo, how epithelial cells form and maintain tubular organs of the proper shape, size, and position, and how organs acquire the capacity for high-level secretory activity. Debbie's also a dedicated and selfless mentor who loves spending time in the lab and with her trainees. When she was inaugurated as the Bayard Halsted Professor last year, the very first thing she said as she came to the podium to accept the professorship was, and I quote, "All this thing about what Debbie did. I just want to point out that Debbie doesn't do anything without a team of scientists working with her." And here she is with her team. She's described by her nominators as having set an extraordinary example as a professor, as a leader of a research lab, and as a colleague, guiding their scientific development, cultivating their inquisitiveness, and supporting their career trajectories. She's also an award-winning teacher and an outstanding community citizen, both at Hopkins and within the fly community. She's served on the Drosophila board in many different capacities, including as president, and has co-organized large international meetings, most recently taking on the role of co-chair of the organizing committee of the Drosophila Crete meeting, the longest running of the EMBO workshops. In a final quote from her nomination package, she's described as a wonderfully positive, interactive, and full of energy member of the community. So I'd like to welcome Debbie Andrew up to the podium and congratulate her. I want you to join with me in receiving the Lifetime Achievement Award for her exceptional, outstanding, and sustained contributions to the field.
It's embarrassing to be up here. It's a little embarrassing to get awards, in part because you're humbled by the amazing things that your colleagues have done, and you realize there are many people who are deserving of these awards. I'm particularly impressed with a group of scientists who got awards from the SDB this year. As is clear, we don't do anything without the people who train us, and we don't do anything without the people who work with us. So I do want to give a shout out to David Kuhn, the person that really helped me pivot from freshwater ecology, in case you didn't know what limnology was, to working on flies, because to me, I had found my home once I was introduced to genetics and development. Okay, I also want to thank my family. My husband is here for 24 hours in Atlanta to support my receiving this award, but he has been there for the last 30 years supporting my career, never ever once putting his career ahead of mine. And so that matters a lot, to have a supportive spouse or other person. Okay, so my decision today was anyway, I decided today that I would mainly focus on science and some of the newer science that's coming out of our lab. So as you may know, the major organ that we have studied in development is the Drosophila embryonic salivary gland. And as a model system for understanding how organs are specified, how they specialize, and then how they acquire the morphologies that are optimal for their function. And we spent a number of years, starting with my work as a postdoc, identifying the factors that are required to specify the salivary gland fate and to maintain it. And then we've characterized transcription factors that are involved in, again, increasing secretory capacity in the salivary gland, and other factors that are involved in determining what kinds of gene products are going to be made by that organ. But today, what I want to focus on are some new genes that function downstream of the FOX8 transcription factor and that play an important role in the final architecture of the salivary gland, making sure that the salivary gland tube has the right dimensions for it to fit nicely in the body, as do all of our internal organs, and to optimize function. So, salivary glands form tubes from, let's see if I can do this, from two plates of cells that are on the ventral surface of the embryo. In a matter of about 6 hours, these two-dimensional plates of cells are fully internalized and form elongated three-dimensional tubes. And this entire process occurs in the absence of cell division and in the absence of cell death, so that the cells that you start with are the cells that you end up with in the end. And this means that there's a limited number of morphogenetic events that can contribute to this organ. So what we know is that the cells can change shape, the cells can change neighbors, they actively migrate as a collective to get to their final correct position in the embryo. These cells grow, and I think that's it. Anyway, so over this period of time, that means that it's a much simpler process to follow, that there are a limited number of things that can occur. And what I'd like to talk about today are the events that occur during the earliest stages of salivary gland formation that determine the final dimensions of the salivary gland tube. So salivary gland internalization begins with apical constriction. And as you see in the images in the top, these sagittal views show that cells in a posterior position within the salivary gland go from having the columnar shape they started with to being wedge-shaped and internalizing. And if you look at the en face views of the salivary glands that are stained with an apical junction marker, what you can see is that the cells that have the smallest apical area are the first cells to internalize to begin to form a tube. Now, the process of apical constriction in many systems, as many of you know, is driven by myosin 2. And in the forming salivary gland, there are 3 different flavors of apical myosin during the early stages of salivary gland internalization. And these include this apical medial myosin, which is pulsatile and is involved in ratcheting of the cell into smaller and smaller apical areas. So that's seen in the cells that are the next to internalize. This is the cells that have already gone in, and you can see this apical medial pool of myosin. There's also this supracellular myosin cable that has been well described by Kacher-Roper's group at Cambridge that forms around the entire salivary gland primordia. And then there's also junctional myosin, which has slightly different distributions in cells near the invagination pit than those cells further away. This process of invagination of the salivary gland is absolutely dependent on the transcription factor FORCAD, which is the single Drosophila ortholog of the FOXA family of transcription factors. And what we observe if we look at stages when the wild-type salivary gland has fully internalized, what we see is that all of the salivary gland cells are still sitting on the embryo surface. I'm having a Mike Levine problem. We're going to go back there. Those cells are still sitting on the embryo surface in the FORCAD mutant. And if we look at even earlier stages when the invagination pits are evident in the wild-type embryo, we see that nothing has happened in the FORCAD mutant. Not surprisingly, FORCAD affects these different pools of myosin that drive internalization. So for example, if you look at the wild-type salivary glands up here, you can easily see the supracellular myosin cable. It's completely absent in the FORCAD mutant. If we look at the apical medial myosin, which is easier to see in these enlargements, what you can see are these blobs of apical medial myosin in the apical domain of the cell, and those apical medial pools of myosin are absent in FORCAD mutants. And even the distribution of junctional myosin in the wild-type embryos looks different in the FORCAD mutants, suggesting that FORCAD affects all three pools of apical medial myosin. So in work that we have previously published, mainly led by a former postdoc in the lab, Se-Yen Chung, we showed that the FORCAD transcription factor drives these apical medial pools of myosin by transcriptionally upregulating FOG, which is the same ligand that functions in the internalization, driving the internalization and apical constriction of the mesoderm in Drosophila. And FOG works through a GPCR signaling pathway to activate apical medial myosin. At the same time, FORCAD also upregulates expression of the Crumbs protein, and through a very elegant molecular pathway, this Crumbs high levels in the salivary gland is responsible for forming this supracellular myosin cable that surrounds the entire gland, again work by Kacher-Roper's group. But it also turns out that Crumbs, when overexpressed in the salivary gland, can expand and stabilize the apical membrane. And so I would like to propose that the process of invagination of the salivary gland is a competition between the forces that are driving the destabilization and shrinkage of the apical membrane, working against the forces that are expanding and stabilizing these apical junctions. So if we look in throughout the salivary gland, in fact, what we observe is that these two proteins show anisotropic localization. So everywhere the myosin protein is high, Crumbs is low, and where Crumbs is high, myosin is low. And Crumbs is mainly localized to the junctions between neighboring cells, where it's stabilizing those junctions and trying to hold the cells together, whereas myosin, both the apical medial and junctional pools of myosin, are destabilizing those membranes and driving the internalization process. So we've recently begun to spend a lot of time on other transcription factors or other transcriptional targets of the FORCAD transcription factors, and those are what I'm going to talk about today. So in a brute force in situ hybridization screen where we looked at over 100 different salivary gland expressed genes, we identified a transcriptional target, a gene known as ARC. And what you can see in the wild-type embryos is that this is expressed abundantly in the salivary gland. The RNA localizes apically. It's also expressed in some other tube-forming tissues. And if we look at a fork head mutant, that expression is lost. The other transcriptional target that we identified of fork head that I'd like to tell you about today is a gene called outspread. And again, you can see that outspread is also localized to the mRNA is localized to the apical domain. And in a fork head mutant, that expression in the salivary gland disappears. Now, not much was known about these proteins. ARC was first described over a century ago during the last pandemic. ARC mutants are fully viable, they're fertile, and they have arced wings. As you can see, unlike the flat wings of a wild-type fly, these genes are arced, and we have recently made complete null mutations of these genes. We see the exact same phenotype. Outspread gets its name because the flies are also viable, fertile, and then have outspread wings. And in fact, this mutant was first described in 1977, and some of the more severe alleles of outspread were shown to resemble those of the ARC mutant. So two transcription factors, two transcriptional targets, apically localized, both have effects on wing morphology. Now, I'm going to show you that ARC and outspread mutants have indistinguishable salivary gland defects. I'm going to not show you, just tell you that ARC and outspread double mutants have more severe phenotypes. And although both ARC and outspread are fully viable as individual mutations, when we combine those mutations, they die. And what that suggests to us is that these proteins function in the same process, but probably do so through separate pathways. So let's first take a look at ARC. So we made null mutations, Let's just keep everybody awake. We made loss of function mutations in ARC. A little disappointed at how normal the salivary glands look. So this is wild type, this is an ARC null. But when we looked carefully, what we found was that the salivary gland tube was shorter. And this is just the quantification. Wild-type salivary glands are about 75 microns long. Mutant salivary glands are now about 20% shorter. And if we overexpress the ARC protein, what we find is that the salivary glands are longer. And this is the quantification of that data. Now, this is independent of embryo length. And so one question is, if cells aren't dividing and they're not dying, how do you create tubes of different size? Well, it made us think that maybe perhaps it's in the arrangement of cells that things are different. So what we did was to look at optical cross-sections of the salivary glands at multiple different positions and simply count the number of nuclei surrounding the salivary gland tube. So in wild type, on average, we find that there are about 7.5 cells in cross-section of the salivary gland. In our ARC mutants, we're seeing that there's approximately 9.2 cells on average. And in our ARC overexpression salivary glands, we find that there's somewhere around 5 cells in circumference. So what this tells us is that the defects that we observe in salivary gland length are a consequence of the arrangements of cells within the tubes. So wild-type cells, approximately 8 cells in circumference, approximately 18 cells long. ARC mutants are about 9 cells in circumference and correspondingly shorter. And our overexpressed salivary glands, cells overexpressing ARC, have fewer cells and are even longer. And in fact, with ARC overexpression, we often see salivary gland cells that remain on the surface of the embryo. So, how does loss or overexpression of ARC affect tube geometry? Now, because we can see differences in the morphology of the salivary glands in wild type versus ARC mutants at very early stages, we wondered whether it was the process of invagination that might be different in ARC mutants versus wild type. And one way you could create tubes that would be of different dimensions would be at this very early stage when the cells first invaginate. If you had a different number of cells internalizing at those early stages, they would then potentially scaffold the tube so that you would end up with either tubes with more cells or fewer cells. So because crumbs and myosin played important roles in this process of internalization, we asked whether we could phenocopy some of the effects of loss of ARC or overexpression of ARC by messing around with myosin or with crumbs. And what we find, again, is that in wild type, you have the longer salivary glands. ARC have shorter salivary glands. Again, if you overexpress ARC, you have longer salivary glands. And what we found is that if you hyperactivate myosin by overexpressing the myosin light chain kinase, we get the same phenotype as loss of ARC. Whereas if we degrade myosin specifically in the salivary gland by expressing a nanobody against a fusion protein, what we find is that you phenocopy the overexpression of ARC. And finally, if you knock down Crumb's function by overexpressing just the intracellular domain of the Crumb's protein, you get phenotypes that look exactly like those of ARC loss. And this is not only in the length of the tubes, but here I'm showing the data for how many cells on average there are in cross-section of the different mutants. So loss of ARC looks like overactivation of myosin, and it looks like loss of Crumb's. So this suggests that ARC interacts in this pathway by supporting what Crumb's is normally doing in the salivary gland. And we would argue that it's stabilizing and preventing the cell shape changes and rearrangements involved in internalization. So how does ARC do this? It turns out ARC encodes a large novel protein which contains 2 PDZ domains, which are the only recognizable feature of this protein. When we look at antibody staining with ARC and Crumb's, what we find is that ARC and Crumb's co-localize in the salivary gland, shown in the merge that you can see here, but also that the co-localization of ARC and Crumb's is limited to the salivary gland. So in this sagittal view, you can see both proteins are localized in the most apical region of the salivary gland, whereas if you look at these cells that are just posterior to the salivary gland and enlarged here, what you see is that Crumb's protein is apical, the little red staining you're seeing that I hope I'm pointing to, and the green staining with ARC is all along the lateral membrane. So it's only in the salivary gland where these 2 proteins localize. Next, we asked if ARC had any effect on Crumb's accumulation in the salivary gland. So again, this is a wild-type salivary gland showing you the very high levels of Crumb's protein within the salivary gland cells. And what we find is that in ARC mutants, the levels of Crumb's go down, as you can see from quantifications here. And if we overexpress ARC, what we find is that we don't necessarily see increases in the amounts of Crumb's, but we see changes in its localization. So in this experiment, we're overexpressing ARC and then looking at where Crumb's is localizing. And what you can see in wild type is that Crumb's is entirely on the plasma membrane, whereas with ARC overexpression, we see Crumb's beginning to accumulate in an apical medial region on the apical surface. So ARC affects not only how Crumb's gets to the membrane, but also where Crumb's localizes in the membrane. And I should mention that ARC is also in the apical membrane where we see Crumb's. So we did a FRAP experiment on Crumb's to ask how Crumb's would relocalize to the membrane if we did a photobleaching experiment to see if ARC had any effect on the rate at which our Crumb's protein localizes back to the membrane. I'm not sure how well these movies will play, but we'll give it a go. We're using white arrows to point out where we're bleaching and deleting the Crumb's signal in the membrane, and the yellow arrows are going to point to where that same position because things move around a little. And what we're looking at is the disappearance and reappearance of Crumb's protein over time. I'm not sure it's playing. We're going to go to the bottom line. So if we look at how much fluorescence recovery we see in wild type versus ARC mutants, we see it's exactly the same. And this suggests that the mobile fraction of Crumb's protein in ARC mutants is no different between wild type and ARC mutants. But what we do observe is the rate at which the signal returns is much faster in wild type than it is in the Crumb's mutant or in the ARC mutants. So somehow ARC is facilitating the localization of Crumb's to the plasma membrane. ARC also affects the localization of myosin 2. And this is just 2 images showing, again, wild type on top, where you can see the myosin in green and an apical junction marker in red. And if you look at ARC mutants, it's probably obvious that things are different, but it's a little hard to see how they're different. And so we started looking at the levels of myosin as a function of how many cell diameters you're away from the invagination pit. So that's just shown here. So if the invagination pit is indicated by this asterisk, we then calculated the intensity of myosin staining as you moved 1, 2, 3, 4, and 5 rows of cells away from where the pit was found. And we quantified separately the apical medial pools of myosin and the junctional pools of myosin. And these graphs show what we find. If you look at wild-type salivary glands and the accumulation of apical medial myosin in cells 1 to 5 cells away from the invagination pit, you see this nice slope where myosin is gradually decreasing as you move away from the invagination pit. When we look at the ARC mutants, we see that levels of myosin are much more stable across many cell diameters. Correspondingly, we see a similar trend with the junctional myosin in that it gradually decreases as you move cells away from the invagination pit. Whereas we see a stabilization many cell diameters away from the invagination pit in the ARC mutants. Now, this has consequences on apical constriction, and this is just showing you a wild-type salivary gland stained with E-cadherin to mark the adherence junctions versus an ARC mutant. Some of you at the front might be able to see that there are cells with very small apical areas that are very many cell diameters away from the invagination pit. Now, we've colorized this so that you can see that in wild type, where blue indicates the smallest apical area, it's very well organized so that cells that have the smallest apical area are the cells that are closest to the invagination pit, which makes sense. These are the cells that are going to internalize next. And then you see the cells that have slightly larger and then larger apical area. Now, this is not true in the ARC mutants. Again, we can see cells that are several diameters away from the invagination pit that have very small apical areas. Now, we're just working to do a more complete mapping of apical area with respect to location within the salivary gland placode. This is just showing you data for the samples we just looked at. And as you can see, the cells that have the smallest apical area, the smallest 10% of cells with apical area, are closest to the pit in wild type, whereas you will see those cells many cell diameters away in the ARC mutants. So the other observation we have is that when you overexpress the ARC protein, you get the same phenotype as you get when you overexpress the crumbs protein, and that's an enlargement of the apical area of cells throughout the salivary gland. So this suggests a model to us where ARC may directly interact with the crumbs protein to stabilize crumbs at the membrane to counteract the effects of myosin. So myosin would be driving the cell shape changes that fuel the internalization of salivary gland cells, and ARC through crumbs is stabilizing the membrane to limit the number of cells that undergo apical constriction. And as a consequence of the number of cells that undergo apical constriction at a given time, in the loss of ARC, more cells would be internalizing, creating tubes that have more cells in circumference and fewer cells along the length of the protein. Now, it's possible that these two proteins directly interact because there are two PDZ domains in the ARC protein, and there's a PDZ binding domain at the very C-terminus of the intracellular portion of the crumbs protein. So far, we've not been able to show that they physically interact using any biochemistry, but it's possible that these interactions require some tension for the binding to occur. Okay, so I've told you all about ARC. We can go through outspread pretty quickly because I've already told you that the salivary gland phenotypes are very similar. So if we look at loss of outspread and compare them to wild type, what we find is that the salivary gland is shorter in outspread mutants. And when we look at the number of cells in circumference of wild type versus an outspread mutant, what we see is that there are more cells in circumference in the outspread mutant. And this is independent of salivary gland cell number. This is just counting the total number of nuclei in the salivary gland. And as I already mentioned, loss of overexpression of ARC or overexpression of outspread give very similar phenotypes, and they're the same phenotypes that we observe when you degrade myosin specifically in the salivary gland. So this suggests one simple model, and that is outspread somehow negatively regulates myosin. Okay, and we're particularly enchanted by that model because of what outspread encodes. So outspread is also a very large protein. It's related to a mammalian protein known as myosin phosphatase Rho interacting protein, or MRIP. This bottom slide is just showing you, or this image is just showing you, that like MRIP, outspread has coiled domains at its C-terminus, and both proteins have N-terminal pH domains. So what's known about mammalian MRIP is that it directly interacts with a protein called myosin, or interacts directly with myosin binding, the myosin binding subunit of myosin phosphatase. So this myosin binding subunit is activated by Rho kinase signaling, and the C-terminus of the protein directly binds to the C-terminus of MRIP. And what it does is to recruit myosin phosphatase that would deactivate the myosin protein by removing those regulatory phosphorylations. So our first question was whether outspread co-localizes with myosin. And what we observe using antibodies against the endogenous outspread protein is that outspread accumulates in the apical region of the cells that are about to invaginate into the salivary gland. If we look at sagittal views of the salivary gland, what we see is that, the bottom here, is that myosin and outspread are at least in the same neighborhood. They're close to one another on the apical surface. We then ask if outspread localizes with the fly version of MBS. And again, these are overexpressed proteins, either tagged with GFP in the case of outspread or with HA in the case of MBS. And what we can see is next to the invagination pit, we can see both outspread and MBS completely overlapping one another's localization. So it looks like the two proteins have the potential to be interacting, just like is observed with the mammalian proteins. So the next question was, do they interact through their C-terminal domains? Okay, Ken's giving me the, okay. Let me just tell you, outspread forms long condensates or aggregates. There are domains within the outspread protein that are required to form these aggregates. The endogenous protein, when we look at the salivary glands, could certainly be forming aggregates in the salivary gland. If we express both the flag protein and the outspread protein in tissue culture cells, what we see is that every time we see them, they're always together. And so we use this assay to map which domains are required. It's the C-terminal domain of the myosin binding protein and the C-terminal domain of outspread that are required. The pH domain is required for the apical localization. Outspread binds probably through its pH domains to pips, and the pip that's present on the apical surface is PI45-PIP2. So we have done a structure localization analysis of the outspread protein. This is required for apical localization. This is required to form the condensates. This is required for interactions with the myosin binding subunit. We've yet to do the structure function analysis of outspread protein. So we're building all of these same constructs, putting them back into flies, and asking how each of these constructs affects myosin activity and how loss of outspread might affect myosin activity. Our working model, based on the localization of outspread and who it interacts with, is that while GPCR signaling activated by FOG drives the phosphorylation of myosin, which drives apical constriction and internalization, is counteracted by the outspread protein, which brings in this myosin phosphatase, perhaps creating the rise and fall of the myosin that occurs during the process of apical constriction. So I don't know why it's animated, but what I've shown you is that this FOXA transcription factor both drives the expression and activation of a pathway that activates myosin, while at the same time activates expression of a factor that, there we go. The guy turned me off. Okay, I'm just going to do it by, okay, maybe I turned me off. Okay. Anyway, it activates crumbs, which counteracts myosin activity, and it also activates expression of yet another protein that limits myosin activity in the salivary gland. So it looks like, so far, all of the targets that we've identified for this transcription factor converge on two proteins, one that's holding cells together and preventing them from changing their shape and rearranging, and another that's driving the apical constriction and cell rearrangement that's required in order to internalize and build a tube out of a plate. So this work was done by several postdocs and graduate students. These are the people that began the work on forkhead, FOG, crumbs, and Methuselah-like thigh, which we think is the FOG receptor in the salivary gland. These people did the early work on outspread and ARC, but this is the person, Jahoon Kim, an amazing research associate who is bringing this story home. So I hope to publish these stories soon so that you can get the slower version. Okay, thank you.